for joining us this afternoon. This session will be recorded. Okay? So wishing one and all keeping safe and keeping well. Some housekeeping. Before we get into the presentation proper, all mics will be on mute. Any questions, please raise it up in the chat box. However, at the end of the session, we will have a feedback form that you can further input your questions, plus a return email address is compulsory so that we could reply to you via email as well. Okay. So this afternoon session is a sequel to the forum held in August 2019, titled The Peranakan Identity, Who Am I? Thank you to all the volunteers who donated and stepped forward with their blood samples for this research. For those who have not got their results, please get in touch with Peter Lee. You have his contact. So it is ready and the results are out. Get in touch with Peter. Okay. Oh, both Professor Roger Foo and Professor Wang Chaolong will share in greater detail on the project's findings, with TPAS President Colin Chi presenting the culture and heritage aspect. An introduction. Professor Roger Foo holds the appointment of Director in the NUHS Cardiovascular Disease Research Program. He is also appointed as Assistant Dean of Research at the School of Medicine, Head of the Scientist Academy, Senior Group Leader in 2019 at the Genome Institute of Singapore. He will speak about the genetic makeup of the Peranakans' DNA. Professor Wang Chaolong is a professor at Huazhong University of Science and Technology. He received his PhD from the University of Michigan. He then moved to Harvard University to conduct postdoctoral research in statistical and genetics. He has done extensive research in studying human population history from genetic data. For this afternoon, he will be sharing with us the project's aggregated findings from the research done. And Mr. Colin Chi, current TPAS president. His maternal side is Sarawak Peranakan. He was a former journalist with the Business Times, and thereafter into the corporate world for 30 years with ExxonMobil Asia. Under his leadership, especially during these COVID years, it has been really tough and challenging. However, under his helm, the hybrid 33rd Baba Nonya International Convention will come to fruit on 20th November 2021. Have you purchased your virtual tickets as yet? So we still have tickets available. This is the EDM for you to purchase your tickets from SysTech. Now over to Professor Roger Foo to kick off this afternoon's presentation. Over to you, Roger.
Thanks, Gwen. It's so exciting the way that you introduced us with such declamatory tones. Okay, let's share screen here. Can I share screen?
Yes, take over the screen.
Okay, I hope I manage. Can you see it?
Yes.
Okay. So folks, thanks so much for bringing Chaolong and myself to your meeting today. For me, I'm definitely very pumped up. And I thought of mentioning this anecdote. My wife says there can be not that many things as exciting as coming on a Saturday afternoon to speak to Peranakans. And I believe that's quite right, because my wife is hardly ever inaccurate. So here am I. So my job is actually to kind of give a foundation before you hear the real stuff that Chaolong will be telling you about. So in my case, I'm a cardiologist with deep interest in cardiac genetics. Because of being a clinical geneticist and genetics specialist, so this puts me in a perfect position to be able to say these things to you as a foundation of what genes are, what variations mean, and then maybe just one paying respect slide to disease before we hand off to Prof. Chaolong, because that's the real meat of what you want to hear this afternoon. Okay, so here we go. Mine is really a much shorter one, and I'm having the better deal between the two of us. So I felt that it was quite a useful opportunity to explain what a gene is to this population, to this group here. Some of you might be biologists and have good knowledge about it. And then you would know, of course, that the gene in the best definition possible is the smallest indivisible unit of inheritance. So depicted here in this box is a gene. There are about 20 to 25,000 genes in our human genome. And of course, again, amongst the biologists of you, you might immediately recognize that this is referring to something called the protein coding genes. So in effect, genes are a string of bases. The bases are the chemicals that make up your DNA. So we're talking really about the DNA. And there are basically at the root of everything, 4 bases in our human genome. So the bases are abbreviated to the letters A, C, G, and T for easy recognition and following. And so in other words, each gene really is just a string of bases. So we are broadening out now. So if you look at genes in our genome, the best way to think of them is that they are lined up in series like this. So you can see the various genes on the string here. And genes can be of varying lengths. Some are longer than others, some are shorter, and so on. And in between genes, you have these varying distances as well. So some of these distances can be really very long, tens of thousands of bases between genes. So if you add up everything together, bases between genes and the bases inside the genes themselves, altogether, the knowledge is rounding up to roughly 3 billion bases, making that one human genome. So by this, you immediately can see that the single human genome is vast. And the analogy that I really liked when I came across this was how they say that this, famous book is made up of 3 million letters or alphabets. And if one single human genome is 3 billion bases, so literally a single human genome is 1,000 of these Bibles. And if you go down to the museum on Euston Road, and maybe there are geeks in this community too who like to do such a thing when they're on a holiday, you will see this very shelf where the human genome is written out or typed out or printed out onto these volumes of books here. So for example, chromosome 11 occupies 1, 2, 3, 4, 5 of these volumes, and chromosome 12, etc. And when you open these books up, you literally see the letters A, C, G, and T spelled out like that. And this really immediately strikes you at how inscrutable this code is, this human genome. So then we need to get to this point about variations, because variations is what really tells us more about the human genome and what makes us different. So medical scientists make use of a single reference because you need to establish, and that's what we have done, what is the basic foundational reference. So there is a reference human genome, which is exactly that genome that is put out in the Wellcome Trust Museum. And from that reference, anything that differs from that reference is therefore called a variation. So if we looked into this, again, back to that genome that I showed you earlier, or that section of the genome, and if you magnify that section out, you will see these letters. For example, this is just very arbitrary example. And a person has G at that position, and that may well be what's in the reference human genome. But in another person, he could be a C instead of a G. And further down the road, he could be another G instead of that A. So these are so-called variations. And variations are important because these would then be the ways in which we tell differences between people. And these are the variations that Chaolong has used to do the sort of analysis that you're going to hear about. Okay, coming to the end of my slides, my last one to pay respects to disease. So each gene confers function to a cell. This is something very standard, and the biology, everyone would accept it. Many genes participate together to produce functions of that cell, and cells together would then function for organs, and organs together would give function for that human body. So when you have variations sitting inside genes, this is when these variations can cause damage to the function of that gene. So if a gene is a very crucial gene, and if it contains a single variation that causes damage to function, this is often called mutations. So there are also other genes that when they are singularly altered will only cause mild change in function or not even noticeable function. So I came across this analogy, and I think it really depicts it well. And that's the analogy of a pipe organ. You could have a situation where an entire pipe is taken out, so the whole organ just falls apart. Or you could have the situation when the pipe is just merely playing out of tune, so the organ still plays on, but you know that something's not quite right. So I think those are the kind of nice analogies to put that all together. And moreover, this is the point to make, to say that there's so much unknown still in our biology and science. We really only know well the function of about 2,000 of these 20,000 genes. So in effect, it's still not all that useful to do gene testing on the broader scale. That's what would be worth just mentioning. And then, actually, that wasn't the last slide. I do have some more to say here. This is a depiction of what in biology we call the central dogma, which is that DNA is the blueprint. It transcribes in the cell to these things called messenger RNA. We are hearing a lot about these in respect to the vaccines these days. And then RNA is translated into proteins. So proteins is what makes up the antibodies, they make up the enzymes, they make up the cellular structures, they give the cell its function. And it turns out that some RNA also do that. So not all RNA is just a message for proteins, in other words, just for the nuanced detail for some of you who want to get into that. But basically, the DNA is the template and the blueprint. And this is why I find that this is hugely relevant in our world today, because we know that there are these various different biologics that can be used, for example, in this case, to produce vaccines and give vaccines, which could be vaccines in the form of proteins, vaccines in the form of DNA, or vaccines in the form of RNA. Okay, so I'm really coming to the end now. So this last slide, it really is the last slide, is to just depict to you where genetics can make a difference or can underpin diseases. And the way to really best think about it is that there are diseases that are common, which is on the left side and end of the spectrum, and there are diseases that are rare. So in the rare disease realm, it's really quite easy to understand. There is one gene, when mutated, would cause those singular diseases. So there are famous ones in our cardiovascular field. Some of you might recognize some of these names: Marfan syndrome, Brugada syndrome, long QT, etc. So these are single genes causing high penetrant diseases. They are classified as genetic diseases, one gene, one disease. So what may be more complicated and difficult to tease apart is at the left end of the spectrum, which is common diseases. These are frequently called polygenic diseases because many genes, many factors come together to give you the disease. Things like diabetes, heart failure, hypertension, strokes, etc. So these are common diseases with factors that are even non-gene related, environment, diet, smoking, exercise, etc. So in some cases, common diseases and rare diseases may look very similar. So if your heart gets dilated, it gets dilated even when it's a common cause. it can also get dilated when it's a rare cause. But the key is that underlying that could be a rare reason, which is that you have a single gene mutation. So this is really too much detail. Human genetics is not why we are here. So we can discuss that another day. This is where I stop because I think this is now the time for you to hear what Chaolong has done with your donated blood samples and shown us the display of your Peranakan genetic ancestry. Chaolong, you're still muted. Gwen, do we go directly to Chaolong? Chaolong,
Okay.
Yes, yes. Yeah.
Okay.
Can you hear me now?
Yep.
Yep.
Yes, we can hear you, Chaolong.
Yeah, yeah. Thank you, Roger, for the very nice introduction about the genetics. And let me share my screen. Can you see the screen now?
Yes, not yet full screen, Chaolong.
Okay.
Yes, now.
Okay. Hello, everyone. It's my great honor to be here again. Actually, last time I was in Singapore in 2019 and seeing you guys in face to face, and this time we are doing this virtual video. But it's so excited to introduce to you the findings of this study. We are talking about the genetic admixture history of Peranakan Chinese in Singapore, and now I'm in Wuhan. This is where I am now. And so Roger just gave a very nice introduction about what DNA is, and this is actually my research focus. And I have been studying this genetic mutation that Roger just introduced. And the question is what we can learn from these ATGC, these different letters in your DNA. Roger talked about from the perspective of the disease, and here I'm going to tell you another side of it that we can learn about the history from this DNA. So this is what Roger just covered, but just one more thing, like some famous example, like when we talk about these genes, one of the famous thing is like pigmentation. The white people, they have lighter skin colors, and the rest maybe have darker skin colors, and this is actually they determined by certain genes, and the frequency actually differs between populations. That the Caucasians, they have a higher frequency of certain mutation, like maybe T allele or something in this gene, and then the rest of the world have the other letters or bases. So across the genome, there are actually 3 billion, and only about 0.1% of them are difference between two individuals. So every two of us, actually, we have very similar DNA, but it's just those 0.1% difference that make us look different. And this actually, when aggregates across the genome, we can actually separate different populations just by looking at their DNA. And this is one of the statistical analysis that we try to analyze the genomes of Singapore populations, Chinese, Malay, and Indian. And on this statistical map, we can see that Chinese cluster together, Indians clusters together, and this is the Malay, Singapore Malays, and they cluster together. And this DNA variation eventually will give us to the phenotype variation that different people look different in their faces and other phenotypes as well. And I work in Singapore I was a colleague with Roger for more than 3 years in Singapore in Genome Institute of Singapore. And so it's a very unique country. It's a small country. Like when we look at the Google map, we always need to zoom in twice in order to see this red dots island, the city states here. But the unique thing about this country is actually has very diverse populations. And on your IC card, you can see that you will have a category of either Chinese, Indian, Malay, or maybe others. I think CMIO classification in Singapore. And so here in this presentation, when I mentioned Malays, actually I'm following the official classification of the Singapore government that Malays refer to the local Southeast Asian populations in a broader sense. And so given this genetic diversity of Singapore, actually, it has a very unique strength is that Singaporeans can provide a very good snapshot of the genetic diversity across Asia. So we can see that it has a representative of East Asian population, which actually have 1.6 billion population size in total East Asian. And then we also have a South Asian populations, 1.8 billion. And then Southeast Asians, that's about 600 million. So in total, this sum up together is probably 2/3 of the all the populations worldwide. So actually, this is the unique thing about this country. We can have a potential impact by studying the genetic variation among Singaporeans. And that's when I was in Singapore in the Genome Institute, we start this project called SG10K. And this is the pilot, our pilot study has already been published in 2019. It's actually a cover article on the on Cell. This is a very prestigious journal in our field. So by studying these Singapore populations, we can see here this in this chart, different color actually represents different ancestry components, and we compare to other databases, like we have South Asian populations, we have East Asian populations. This is where they locate these different populations. And here is our Singapore populations. And we can see that these Singapore Chinese actually it comes from China and represents all these, have the ancestry component of all these East Asian genetic components. And then these Singapore Indians represent these South Asian. And Malay, Singapore Malay actually has its unique ancestry component, this light purple color, and the orange color actually is a Southeast ancestry component that is quite unique. And this is how Singapore populations can have a good snapshot of the Asian population genetic diversity. And in this country, or more broader in this area, like along the strait, we know that Peranakan is also the unique population or the unique community in this unique place that it actually has a very famous, well-known mixed culture. And we can see from these food, dresses, and other cultural elements that we can see a lot of the Chinese traditions with elements from the Malays, and also the languages is a mixture. And these are the things I learned about your community when I search on the internet, and I also experienced this very tasty Peranakan food when I was there. And but the Peranakan, whether it's genetically or the mix or not, this is when I was there, I heard from my colleagues, and then when I talk to people, actually, it's actually controversial. That some people believe that they are pure Chinese without a mixed blood, but the others actually will disagree. And I also find that there are different versions about the origin, like the princess story that talking about the Peranakan community are the descendants of the Chinese princess that come to this region in the 15th century. And then there are more generally accepted or more widely accepted theory about the descendants of the Chinese traders that come to this region around the same time. And there are also different kinds of Peranakans, but in our study, we focus on the Peranakan Chinese, and this is the thing. So when it comes to whether it's the mix or not, it really comes to what I have been studying for many years, back to the time when I was in the US. So then I talked to, I have this idea that why don't we just collect the DNA samples and then study from the DNA. And I talked to Roger, I talked to my friends. So basically, this is how we can study the mixture history of the DNA. Is let's think about the mixture is like the first generation. If there is a Malay and a Chinese, when they meet together and they have their first generation offsprings, then we know that your DNA will be half from your father, and then the other half will be from the mother, right? So every chromosome you will have two copies, one from father, one from mother. So then the first child will have one copy that is orange color. This may be the Malay origin, and the other copy will be pure Chinese origin. This is the first generation. You have 50%, 50%. And then from generation to generation, this entire segment will be break down by something called recombination. Basically, from generation to generation, there may be crossover on your chromosomes that break down these segments into small segments to form this mosaic pattern. And when these generations pass down, that it will break from a long check to these small checks. So when we try to infer the history from the DNA, basically we try to color the ATGC letters that Roger just showed you, the book. We try to put colors on it, saying that which segment is a Malay origin, which segment is a Chinese origin. And how this can be done is that from the very first slide I show you that different populations may have very different frequency. And then we just look at the segment and see whether this looks more like to the Malay DNA or more like the Chinese DNA. There will be a statistical method or computational tools that we can infer this, whether it's from Malay or then it becomes from the Chinese side. And once we get this, we try to infer back in time how many generations that this origin or the mixture event Can you hear me?
Yes, Chaolong, you went off for a while. Okay.
Oh, okay. And then, yeah, the method was published by other scientists in 2014 in Science, and this is a well-established method. So then I talked to Roger, and we start these collaboration projects in the summer of 2017. And this is the picture that when Roger was taking the blood sample. And then once we take the blood sample, we also have this questionnaire taking some basic information like your self-reported ancestry, and your blood sample will be processed by professional scientists in the Genome Institute. At that time, JJ Liu's group helped us do this DNA preparation. And then I'm the guy sitting in front of the computer doing the analysis and also do the interpretation. And this is how we work together on this project. And several years ago, and if you remember, this is the flyer that we used to recruit the volunteers to participate in our project. So basically what we looking for, basically we say, if you are Peranakans and you are Singaporeans, and we don't have any further restrictions, except that there is an age range from 15 to 99. And then everyone who self-declared as Peranakans, they are willing to come to participate in our research. They will come to NUS, and then we take the blood sample, do a short questionnaire, and that's how we collect the DNA. And so the purpose of this study is we are not trying to define Peranakans. So we send out the flyer. The participants define themselves as whether they are Peranakans or not. And then we analyze all the samples we collected in the recruitment. So the Peranakans were defined by the participants themselves. And what we are trying to do is we are trying to understand this Peranakan community, trying to understand their population history from a genetic perspective. Some of the few questions we listed is that first is whether the Malays have genetic contribution to the Peranakans. And when we think about this, we can think that if there is a Peranakan community that back in time and along the history, like several hundred years along the way, whether at a certain point, this community have some interaction with the local Malays. And then if they have some interaction, basically they have a inter-ethnic marriage, and then the Malay genes will get into the Peranakan community, will pass down from generation to generation, and we'll try to estimate
Chaolong, you got muted again.
Yeah.
Someone might be pressing on the mute button to mute you, Chaolong.
Okay. Okay.
Okay, now you're back now.
Yeah.
Okay. So we're trying to understand to what extent the admixture proportion is, and then if there is admixture when it happens, and also whether these Malay genes, the Malay bloods, they were contributed by females or males, and these are the questions that we can answer from the DNA, and this is what we have in mind, and these are the purpose of the study. So just bear in mind that this is not a study that trying to define Peranakans using genetics. This is not what we are trying to do. Basically, we are just trying to learn about the history and from a genetic perspective. So we basically collected 177 samples, and you can see that not all of them are Peranakan Chinese. There are self-reported Peranakan Eurasians, 3 of them, and one person self-reported as Peranakan Chinese / Eurasian. And then there is also 1 Peranakan Indian, 1 Peranakan Caucasian, and 5 of them just don't want to answer. And never mind, we will find out. So and there are some related samples in the data. So we remove some of the related samples. Basically, when we try to infer the story, we analyze everyone, but some of the analysis has to be done on unrelated samples just for the statistical analysis purpose. So we compare to some reference populations that Singapore Malays, and then Singapore Chinese, Singapore Indians, and also we also bring in some of the European populations from existing database, so that we can compare to these reference populations to infer the ancestry, the genetic components from different sources. And when we try to put all these 177 individuals into this reference ancestry map, we can see that these red crosses and the blue crosses, these are the 177 Peranakan samples, and they actually cluster closely to the Singapore Chinese. This green color is the Singapore Chinese, and a bit far away from the Malays, Indians, and Europeans, except for some of these outliers. And these we have some statistical definition to say these are outliers to the majority. And we can see a few examples of how they look like. So this is a further close. Basically, this is a 3D map, and then we remove the Europeans and then only look at 3 sources. This is Chinese, Malay, and Indian. And then these are the red crosses and blue crosses. These are the Peranakan samples. And we can see pretty much Chinese, but if we look a closer look, basically we zoom into this region, we can see that Peranakan Chinese and the Singapore Chinese, they are not exactly the same. Like there is a shift of the Peranakan Chinese overall to this Malay Malay population is this direction, right? So we can see that some of the samples actually shifts towards the Malays. And just for comparison, I also put some of the Chinese samples that collected in China. These are Northern Chinese and Southern Chinese. I put them on the same, project them on the same statistical map, and we can see that the Southern Chinese is pretty much similar to the Singapore Chinese, and this is the Northern Chinese group, and this is the Peranakan Chinese. So this is an indication of that the Peranakan Chinese might have some Malay genetic component, but the component, the fraction will not be very high, because if you have very high Malay component, then the Peranakan Chinese is likely to be sitting here rather than here, right? So this is a further look. If we zoom in, these are the Northern Chinese and Southern Chinese and Singapore Chinese, and these are the Peranakan Chinese. So Peranakan Chinese compared to the other Chinese group actually has a shift towards the Malay population. This is from the very broad view overview of this population. And then we look at a few individuals, trying to see the individual, how they look like. We just pick one. from here, this is one Peranakan. In the PCA map, the 3D maps, it is here. And then we look at its genome. There are 22 autosomes, and then you have the X chromosome. This is the sex chromosome, determining whether you are male or female. If you have 2 copies of X, then this is a female. So we can see that this Peranakan Chinese is self-reported Peranakan Chinese female, and from what we can see is a majority of her genome is Chinese, indicated by this light green, but some of these dark green, they are Malay ancestries, is scattered across the whole genome. Sum up together is about 6.2% Malay, and this is actually quite typical along all the Peranakan samples that we analyzed. But we also have some of them, like this one, it's moved. If you compare this one, the location moved from here more towards the Malay, right? So actually in its genome, we can see that more Malay ancestry component. The fraction is about 20% sum up together. And this is one individual that has a higher fraction of Malay ancestry in its genome. And then we also have some Peranakan Chinese that we rarely see any Malay ancestry. This is like 100% Chinese for this particular individual. And we also have very complex admixture, a multi-way admixture. But this person, this individual actually self-reported as a Peranakan Eurasian. So actually we know from the questionnaire that he probably has very complicated ancestry mix up, right? So we can see that they have Chinese, 17%, and then 24% European, 32% Indian, and then 27% Malay. And this is how his genome looks like, very complicated. And in the PCA map, it will be in the center because it has roughly 25% from each of these 4 sources. So it's right in the middle of this map. And this is a self-reported Peranakan Indian. And it's a male because we see on the X chromosome, there is only 1 copy. On the other chromosome, there will be 2 copies, right? So this is a male self-reported Peranakan Indian, and its X chromosome. For a female, the X chromosome of a male will be inherited from his mother. So this is entirely Chinese X chromosome. So this tells us that his mother is actually a Chinese mother. And then the other half of its genome is like almost purely Indian. So this is like one chromosome, half Indian, and then the other half Chinese. And every chromosome is like this. So it's pretty clear that this is a child of an Indian father and a Chinese mother. But the self-identification is Peranakan Indian. And this is what we see in the genome. And it's in the middle, right? This is the Chinese and this is the Indian, and it's right in the middle. And this is and I think this is the only self-reported Peranakan Indian that we have in our 177 samples that we collected. And so just now I just give you a few examples. For every individual, we are going to have a pie chart. And if we are going to another way to draw this pie chart is we just put them into a bar, and then this bar sum up to 1, and the length of each color represent the admixture fraction, right? And by this way, we can put all 177 samples together into this plot. So from high Malay ancestry to low Malay ancestry, these are the non-outliers, and these are the outlier samples. We can see for Peranakan Chinese, the high is probably about 20%, and the mean ancestry is about 5.6% Malay ancestry. But this is a spectrum from 20 to 0, and probably about 10% of the individuals using our analysis method, we don't see the Malay component. But for these parts, even though we see some Malay component, the fraction is pretty low. And this part, we see a lot more Malay component. And this is the autosome. Basically, it tells you the overall picture of the genome. But then we can also look at the X chromosome. X chromosome meaning that the females will contribute more because every female has 2 copies of X chromosome and the male only has 1. And we can see on the X chromosome, the Malay ancestry will be higher comparing to the autosome. So this actually is an indication that the Malay ancestry contributed to the community will have a sex bias, meaning that the more of the Malay component might be contributed by Malay females rather than Malay males. That's why the X chromosome will have a higher Malay fraction. And yes, so this is a sex bias of the mixture. It basically means that the Malay females and the Malay males doesn't contribute proportionally to the mixture events. And this is so this part, the first column is just this one. So I just put them here as a reference, and then I try to compare the other Chinese group. So if we have a comparison, it will be more clear for us to see whether the Peranakan Chinese differs from the Singapore Chinese in general, or the Chinese in Southern China and the Chinese in Northern China. So we can see that these are the general Singapore Chinese is about 1% Malay. The mixture is a very small fraction of individuals that they carry certain Malay ancestry component. And I have to say that this fraction it's possible that they are also Peranakan Chinese, because these are the samples that we have in our SG10K projects. And in the general recruitment of the SG10K projects, we don't ask people whether you are Peranakan or not. It's just they are Chinese and they self-declare as Chinese, and then we collect their samples, we put them into our database. And so there might be Peranakan Chinese in this group. And these are the Chinese in Southern China, and these are the Chinese in Northern China. They are definitely not Peranakan Chinese. So in Southern China, we can see that it's about less than 1%, and Northern China is even smaller. So these are pretty much pure Chinese, except in Southern China, there is maybe 1 or 2 individual that has a higher. This might be due to some of the migration, I believe. Maybe some of the Chinese in Southeastern China, they move back to Fujian or Guangdong, and it's possible, but we cannot tell from the data. And this is the X chromosome. We can see the same pattern. Actually, comparing to the other Chinese group, Peranakan Chinese will have higher Malay ancestry component across both the autosomes and the X chromosome. And statistically, this is a significance. Actually, it's highly significant. Although the fraction is small, but we can see the difference is quite obvious. So statistically, it will be significant. And then we have been talking about this sex bias of the mixture. Actually, there is another marker, another 2 markers that will be very informative about the sex bias of the mixture history. One is the so-called Y chromosome, because we know that Y chromosome it determines the sex, right? If a male, then the sex chromosome will be XY. And if for a female, the X chromosome will be XX. So this Y chromosome is only carried by the males and is paternally inherited. If you are a male and you have a Y chromosome, your Y chromosome must come from your father, because your mother doesn't carry the Y chromosome. And then there is another marker called mitochondrial DNA. These are the mitochondria is in this egg. So when the sperm insert the nuclear DNA, basically the sperm carries very little mitochondria. So most of the mitochondria is in the eggs, but that is you inherit it from your mother. So these are the 2 sex-specific markers that one only inherited from father, the other only inherited from mother. So we can look at the Peranakan genomes, just look at Y chromosome and mitochondria and compare with different reference groups to see what the proportion looks like. And this is just a global distribution of the mitochondrial haplogroup. We can see that there are different haplogroups of the mitochondria, and then different populations worldwide will have very different proportional composition in different populations. And how does it look like in Singapore that we have these Chinese? These are the mitochondrial haplogroup proportion. In Chinese, we can see there are a lot of haplogroup D, haplogroup F, and haplogroup M, haplogroup B. These are the prevalent haplogroups. And in Malay, we can see there is a very unique haplogroup called haplogroup E. And this haplogroup E, we only see it in the Malay population, not in the Chinese and not in the Indian, not in the Singapore Indian. And then we look at our Peranakan Chinese, we do see that there is a substantial amount of haplogroup E, and this actually provides us very strong evidence that the mitochondria carried by the Peranakans, it has a strong indication of that this can come from the Malay females. And this is actually a strong evidence. And from this fraction, we get do the statistical analysis. We have estimates that actually the Chinese probably contributes about 87% of the females, and the Malays probably contributes about 12% of the females to the founders of the Peranakan community. This is estimated from this mitochondrial haplogroup. And the Indians is not significant. We estimated 1%, but statistically, this is not significant. And then we can also look at the Y haplogroups to see the male contribution. And this is in general how the Chinese population looks like. And this is the Peranakan Chinese. We don't see very difference between these two groups, right? And actually statistical test tells us the Peranakan Chinese Y haplogroup composition proportions actually are quite similar to the Singapore Chinese, not significantly different. And if we do the estimate, we can also estimate that the majority of the Y haplogroup probably contributed by the Chinese, and Malay may contribute 5%, but this 5% again is not statistically significant. It can be ranged from 0 to 9%. So we can see that majority or almost all of the male contributions will come from the Chinese community. And I think this is consistent with the patrilineal community in both Chinese and Malay, because when if a Chinese male and a Malay female get married, then this family will be into the Chinese community. If a Malay male and a Chinese female get married, then this will probably absorb into the Malay community, right? So this, I think, is consistent. So now comes to the part that we are trying to estimate how many generations ago this the mixture happens, right? And you don't need to understand these equations, but I'm just showing them here to say that's how we estimate this is we'll fit the data. Basically, there will be a curve, and then we'll have certain models, and then we'll fit the curve. And what this curve tells us is that if this curve decays very fast, then it means that the mixture will be an ancient mixture. If this curve decays slowly, then the mixture will be a more recent mixture. So all you have to bear in mind is look at how this curve, how fast it decay. So then let's look at the data. So I also put Peranakan Chinese to compare with the Singapore Chinese. And if we look at the Peranakan Chinese, there will be a curve that decays. And when we try to fit the model, we have to fit that there will be two mixture events. And we can see the decay speed is actually slowly compared to the Singapore Chinese, right? The Singapore Chinese decay very fast, and then this one decays slowly. So there will be difference between the Peranakan Chinese and the Singapore Chinese comes to their the mixture events. So actually the model will tell us that the Peranakan Chinese will have two the mixture events. One happens about 1,700 years ago, and these are the mixture events. I think is back to the time when your ancestors are still in China, right? It's 1,700 years ago, and the other mixture event will be between two populations, and the two population, 25% will be one source population that have some Malay ancestry or Southeastern ancestry, and the other population will be more like present-day Chinese dominance. And the reasons of the mixture events, this is the one that is unique to Peranakan. It's about 10% that is a highly Malay-like source population. And then the other is more Chinese dominant population source. So this happens about 190 years ago. And if we look at the general Singapore Chinese, and we only see one ancient or the mixture event that we analyze from the Singapore Chinese. And these events, these two analyses, they were done separately. So these are independent analysis, but we can see the ancient or the mixture events, they are actually highly similar. The dates, the timing, they are similar, 1,600 to 700 years ago. And the mixture, the source proportion, they are also highly similar. So this basically indicates this is a shared ancient or the mixture events. And then this is a unique events to the Peranakan Chinese. So this is sums up what I just told you. And putting this together, actually, what I didn't mention is that in our SG10K study, we also have studied the mixture event in the Malay, and we actually infer that the Malay also, the present day Malay, their ancestor also experienced the mixture event about 1,700 years ago of East Asian populations, and more probably at that time is more like indigenous population in this region. So this happens about 1,700 years ago. This actually coincide with the Austronesian expansion. That's the Austronesian people moving from Taiwan all the way to colonize all those islands in Pacific and the Indian Oceans. So putting all this story together is that we think that's about 1,600 years ago, the early Han Chinese from the central China, they actually moved southwards to the southern part of China, and then mix with the early Austronesian people there to form the present-day Southern Chinese. And then the present-day Southern Chinese, when they move to Southeast Asia, they meet these Malay people there and probably have some other mixture events. And the contribution is about 90% to 10%. And this Malay itself is also a mix. I think currently in the present world, everyone is a mix just when it happens, right? So Malay is a mix, Chinese are a mix, and Peranakan are a mix between Southern Chinese and Malay. This is when I say Peranakan Chinese are a mix, I didn't mean that every Peranakan Chinese are a mix. What I mean is that this population in the history, the population actually interacts with the Malay population. So some of the Malay genes was introduced to this community, but not everyone in this community will need to have that Malay component. Some of them will be affected more, some of them will be affected less. That's why there will be a spectrum. So this is what we find. And I think these ancient or the mixture events in the Southern Chinese actually coincide with the settlement history of Han Chinese in Southern China. I think in Eastern Han Dynasty to the Tang Dynasty, there were a big migration of Han Chinese due to the wars. They actually migrate from the Central China to the southern parts. And I think this is what the ancestor of my family. I was Hokkien, right? So my family actually moved to Hokkien around that time. My ancestor, not my family. That's far away. So that I think that summarizes all our findings. And now our study has been officially published in this journal, Molecular Biology and Evolution. This is actually a flagship journal of the Society of Molecular Biology and Evolution. And this is the first page of our study. And very nice to have it published after so many years work. And it's a tough time to get it published, but we are glad that it is accepted by the science community. And these are the research team that I think we took this picture probably 4 years ago, 3 to 4 years ago, that when I was still in Singapore. Many of these are the team members in Roger Foo's lab who helped collect the samples. And these are the postdoc in my lab who did the analysis, and these are the GIS team who help us with the lab work. And thank you for the Community Genome Institute, NUS, and Peranakan Museum, and the Peranakan Association, and my home institute now. So thank you for all your support. I think that's all my presentation.
Thank you, Chaolong. Over to you, Colin.
Okay, hi everyone. Thanks for making an appearance. I'm quite sure you found the presentation by Chaolong and Roger very interesting, and it certainly has stirred a lot of interest and comments in this. I'm not going to make any presentation on the culture whatsoever. I'm just here to present myself as part of the 3-person panel to field any questions that you may have. Chaolong and Roger obviously will be fielding the more science-based kind of questions that you ask. I will attempt to handle the cultural aspects of it and the ethnic aspects of it, the non-science part of it. But I would certainly urge all of you to try to focus on the science part of it, because as you would agree, there are so many questions that can be asked of Chaolong and Roger. And I would certainly want to encourage them. And one thing that I would like to pose to them is a question that was asked us when we first announced the webinar this afternoon, this afternoon's webinar. And that is, you talk about the significance of percentage in DNA, right? I mean, the Malay, on an average, you say that the Malay is pretty low. And in terms of the sample size, people are asking, is 177 a good enough sample for you all to decide on the veracity of the findings, basically? How valid does that make it? The very fact that it has been published in a very distinguished publication would suggest to me as a layman that, yeah, it is. But certainly I would like to hear from Chaolong, who's a genetic statistician and a globally acknowledged scientist in that respect. Maybe Chaolong, you can elaborate on that a little bit.
Okay, so I think comparing to the population genetic studies we did, 177 actually is more than enough. The key question is that about when we recruit the samples, do we recruit it in a biased way? If we don't recruit it in a biased way, the sample size is sufficient, right? So what I mean that if we recruit in a biased way is that we only look at, say, if we look at a person's face and whether it has already indication of certain other mixture, and then we recruit this sample, this will the so-called biased recruitment. And the way we try to recruit is just we pull out the advertisement, and then people who define themselves as Peranakans come to this interview and sample donation. So I think in this way we should be fine in to get any bias analysis results. So I think 177 is good enough for us to get. Yeah. And the other thing to bear in mind is that although the individual is 177, Roger has already shown you that every individual actually is 1,000 Bibles, right, in your DNA. So that's actually a lot of information.
Yeah. Thank you. Gwen, are there any questions from the chat?
Oh yes. Okay. I think I've sent one question to Roger. Will be up his avenue. This is by Mark. How do you label Peranakan Dutch? Only when there are Luso-Malays and other mixed groups which make up this sort of Peranakans.
Gwen, I'm not sure I'm the qualified to answer such a question. The term Peranakan Dutch even sounds very unfamiliar to me. I think it's Colin. Colin, your comment on this label of Peranakan Dutch.
Yeah, I think we also used to I'm no expert in this area. I'll be upfront about it. But having said that, you talk about Peranakan Chinese, you talk about Peranakan Indians, you talk about Jawi Peranakans, and there are also Peranakans among the Myanmar. They have a particular reference for that. And it's therefore not even surprising that we, you know, the Dutch occupied Indonesia for the longest time. And many of the Indonesians, and there were obviously intermarriages in Indonesia, and many of the Dutch and their family members, mixed blood probably, who would have gone back to the Netherlands, so to speak, and they continue with the culture in a way. And there are some marvelous videos that we see offhand where you have groups of Dutch-looking guys singing beautiful Indonesian songs, you know, Dongdang Sayangs and Keroncong and all that. So there is actually a living Dutch Peranakan, if you like, Dutch Peranakan community there, and I don't see any reason why. Recently, I even read something about a research being done by a university in Malaysia. I don't quite get the name, but they are saying they're doing research into Peranakan 6, as in SIKHS, you know.
So what I would say,
that's very
Colin,
interesting.
yeah, and Gwen, is, I mean, for me and Chaolong, this has been as much as anything a learning journey for us, right? To discover that, you know, the term Peranakan is actually itself mixed with a lot of other ethnicities too. So like Chaolong emphasized, and he really took a lot of emphasis on the fact that we invited participants on the basis of their self-declaration. So, you know, we were not setting out to say, you know, define and say what makes up a Peranakan Chinese or what makes up a Peranakan Dutch and all the others. We were literally broad church, Peranakans, come forward. We want to see what your genetics look like. So it turned out that the bulk of the ones we got are people that declared themselves to be Peranakan Chinese.
I think it's very important to remember that what the genetic study does is to provide us with just a metric. And then we just translate that metric, you know, and draw some inferences. And it's so interesting that now we have something scientific, a scientific finding to say, hey, all these self-declared Peranakans who have offered themselves as guinea pigs, if you like, for this study, the large majority, and that's 90% of them, actually have mixed ancestry, you know, Chinese Malay or Malay Chinese ancestry. And it's then for us to say, well, that confirms what my family history, for instance, although I must add that in my own family, or my in-laws, they have a family chupu, which is patrilineal, and they say there's no Malay name for generations. There's no Malay name, and therefore must be pure Chinese. And I think there was a question that was posed a question that was received from one Amy Low. I don't know whether she's around this afternoon, but she asked an interesting question. And what was the question, Gwen? I think it was.
Yeah, regarding the. Okay, I'll get the question. It's on the email.
Yeah, and she talked about mixed ancestry and how it works, and the fact that her great grandparents, I think I had a quick look at it, insisted that they are all pure Chinese, and presumably it's not just family history, also from their own family chupus. And then here you are, suddenly confronted by your own gene statistic that says, hey, you're mixed, man, you're 80% Chinese and maybe 20% Malay. My wife, Linda, who comes from parents who are actually Peranakan all the way through, her mixture is 60 plus Chinese, 20 plus Malay, and just under 10% Indian. Now, where the Indian came from is also something else, you know. So it's so very interesting. So it is just for us to realize that this is a metric that we have, a scientific metric, and then we make our own inferences now. You know, suddenly she realized that, hey, you know, unlike what the family chupu says, she's not 100% Chinese. she's even more mixed than that. She's got dominant Chinese, significant Malay, and equally significant Indian, because you say, right, anything that's above 1% is significant. You know, anything below, yeah, you can just figure that it's not as, it's just nominal, if you like. But anything above 1% is significant. So that's interesting.
Yeah.
I do.
The question I got it is, given the unique history of Malacca, do you think a third box of European should be added to be consistent with its history of colonization? These male soldiers came around 100 to 500 years ago. So it will be about 300 years before the Southern Chinese influx to Southeast Asia. And if so, what sort of percentage would you expect in the Peranakan Chinese from Malacca? I guess she's talking about the European influence here in Malacca. So this is the reference to her question. So she's narrowing it on Peranakans in Malacca specifically. Yeah. So
I guess in our data, we actually have done the analysis that we have a reference population of European. Although it's not Dutch, I think the European reference, what I put in is actually Central Europeans plus some British, but it's a good approximation, a good proxy for the European ancestry. And we can see that at least in the non-outlier samples, some of the outliers has very significant amount of European ancestry, but for the majority of the Peranakan Chinese, the European ancestry is actually quite a small fraction. So it will be very difficult to get an accurate estimate when the mixture fraction is so low. Yeah.
Yeah, that brings us to the next. Sorry, Roger, you'd like to?
I just wanted to ask, because it brings to mind another kind of science maths question for Chaolong, actually.
Yeah.
Chaolong, the analysis of the time decay.
Yes.
You came up with 10% of Malay. But in the earlier analysis, the average percentage of Malay among the Peranakans is 5%.
Yeah, 5.6%. So actually, I think these different statistical models and different methods, when they have based on different assumptions, the estimates will be slightly different, right? From the decay curve, we have an estimate of 10%, and then from the mitochondrial haplogroup, I actually estimate the female contribution is about 12%. 12% female. If we think about that, female contributes half. Like in total, female contributes half, then 12% female Malay will be turning into a 6% total contribution, right? So the 6% is quite consistent to the 5.6% that I showed previously, and the later estimate about 10%, I think is higher, but I think in general, they are consistent. So the conclusion is that there is definitely some Malay contribution, and the fraction is not very high, and it can range from maybe 5% to 10%.
Okay.
Yeah. Just don't take the exact number so seriously. It's a rough range.
That's an important point, I find, because that is a question that is frequently asked from each of the individuals.
Yeah. Yes.
Okay.
Yeah.
So we move on, because I think this topic has generated a lot of interest on people wanting to go to a reliable commercial test center to have their DNA tested now. So they're asking for any recommendations, or how do they continue with that? So there's a generated interest in wanting it to test their blood now. Any advice here?
Yeah, so I don't know whether Chaolong would want to say something here, but for me, I would say there isn't anything out there that would be reliably with a kind of reference to tell you your percentages of Peranakan or this kind of analysis that you saw Chaolong do.
Yeah. So the commercial companies, I think they have quite a number of different products there that they will do the ancestry analysis for the customers, but their analysis, I think, is more like a broad scale analysis. They don't have a specific product for analyzing the Peranakan ancestry. Yeah.
Okay.
Which
So
is
we go back to another question with Malay referencing. When the research refers to Malay, does it mean the residents of the Malay archipelago, that is present-day Indonesia as well as Malaysia? Do you include the archipelago? Or it's just Malay within Singapore context?
Oh, that's a good point. I can jump in here first before Chaolong does the details. So we did, in fact, try to recruit Indonesians as much as possible, again, self-declared native Indonesians, and even taking some guidance from expert within your community of I cannot remember the exact names, like Batak and so on. So we did have a few of those genomes that we could have referenced and looked at, but the numbers in the end turned out to be too small to give themselves sufficient rigor. So I think what Chaolong will probably tell you is that we ended up using the Singapore Malays as the biggest reference, right, Chaolong?
Yes. So I think we have about 500 individuals as the Malay reference, and the vast majority of them are the Singapore Malays. We have a very small number of the Indonesian Malays in it, but I think Singapore Malays, when we do the SG10K project, or also in Singapore in general, we don't have the information about specific Malay group. We only have their self-identity, say Malay, and then that's what we have.
Right. So, okay, there's a lot of interest in this aspect. Another one, if it's possible to determine which Malay group, the Balinese, Batak, Javanese, or Orang Asli, I think, Roger, you've taken that question already. But there's a second part to it. Is there a possibility of early generations being pure Chinese, and then intermarriage only happens at in later generations? Can we identify which generation did they become Peranakans when that intermarriage happened?
How long? Go for it.
So I think the question already set a trap there, that it's not because they have intermarriage and then they become Peranakan. That's not what we're trying to say. So, but we do, like in our analysis, if you remember, that's at the very beginning, I do exclude a few number of individuals that I call the outliers, right? And among the outliers, the number is actually higher than the self-reported Peranakan others, like Peranakan Indian, Peranakan Eurasian. There is also some number that has a significantly higher fraction of Malay ancestry in it. So those samples were excluded, and I think the reason to exclude these few number of individuals is that they are very recent of the mixture. If we think about it, it's a one generation of the mixture, then the participant will have 50% Malay ancestry. If it's two generations back, then that will be 25%, right? So actually, this very recent of the mixture will have that within 1 to 2 generations, they will have much higher Malay ancestry in their genome. And just to be representative, or just to reflect the earlier the mixture history, when this Peranakan community is forming, we focus on the majority that do not have such outlier the mixture fraction. Yeah.
Right. Thank you. Okay.
Can I pose a question? Can I just go ahead and pose a question? You know, so let's say my wife has this, or at least I have this particular genome or DNA profile of 70% Chinese and 20% Malay. Would my siblings have the same profile? Or what, if there's any variability, how wide would that variability be? I think this would be one question that will help answer some of the questions that we have, because maybe their brother has donated blood and but they haven't, you know.
So your full siblings will have a very similar percentage to you, because both of you are inherited from the same parents, right? but your half sibling or your cousins, they might different. For example, if your cousin has a Malay mother, then that will be totally different, right?
Yeah. Okay. But how then do you explain when you talk about genome, it affects your looks as well, right? It affects your looks, person's looks. Does it? Because as I mentioned earlier on.
Colin, you're wondering why siblings can look so different.
Exactly. One looking like a Chinese, one looking like a Malay, another looking like Malaysians, and they come from the same parents.
Yeah. Okay, so that's a really tricky one. So I think the reality is that not everything about us is just down to the genetics. The genetics is the background, but what happens from the moment you are born, and your exposure and the things you do. So this is the environmental influence. It's built upon the background of our genetics. So for example, if you have that sort of genetics, then it will make you more prone to some feature in your body shape and so on. Whereas another genetics, those kind of body features and shapes will never happen in another person, let's say. But the reality is that the genetics in your full siblings, like what Chaolong is saying, should be very, very similar between brothers, real brothers and real sisters.
Yeah.
So that's when you don't want to find any difference.
Well, Roger, I can also add to this. So Roger just mentioned about the influence of the environment. This is another layer of influence in addition to genetics. But even the genetics, even that your full siblings have a similar Malay ancestry proportion to you, but when it comes down to a particular gene, say for example, when it comes to the gene that determine your skin color, the particular gene can be different because and then that's what makes your full sibling, your brother, have a different phenotype, have different looking from you.
Oh, okay. Yeah, then Chaolong, that does add the complexity because each one of us carry 2 copies of the gene.
And the 2 copies of the gene,
which is from our own parents, right? And so, one of those copies goes down to the sibling, and that sibling may inherit the other copy rather than the one.
I see. Okay.
So, which brings me to a very interesting question somebody posed about all these genes and chromosomes. Would the Y chromosomes of the Peranakan X mixture die out eventually? Will the whole Peranakan culture or us as Peranakans die out because we don't have any more of the chromosomes?
I hope that person is not implying that the maleness in your community will fade, right? Actually, I don't quite get the question in that case. The Peranakanness of the Y chromosome?
Yes, that's right. Correct.
So I think the Y chromosome is not unique to Peranakan, right? We see the Y chromosome of the Peranakan Chinese are very similar to the general Chinese. So don't worry, it won't die out. There is a lot of
That's good to know. That's good to know. And then, well, we're almost towards the end of our
Maybe
session.
the way also to flip a question for you, Chaolong, will the mitochondria Malay ancestry in the Peranakans fade away because if they don't have more Malay admixture going forward?
I think this is evolving, right? It just depends on, I think nowadays, I think back in the old time, Peranakans probably have this marriage within the Peranakan community. And nowadays it's probably more like open, right? It's like
also if you don't Peranakans don't intermarry.
So in this sense, I think there will be a lot of other contributions to the community. So it just comes back to the end that this community is not defined by the DNA, it's actually defined by the culture, right? And 200 years later, the study will look different.
Therefore, we have other questions that would like both Roger and Chaolong to go deeper into studies, into further genetic composition of Peranakan communities across Southeast Asia. They're asking you to further drill down on the identity of the genetic makeup of the larger groups, example, Malay, Chinese, Indian, Europeans. You see, so wanting you both to get out there and cover more communities and identifying them as Peranakan. So any thoughts that you will continue with this research in the near future?
So it's always very dangerous, I feel, Gwen, when you ask academics whether they want to do some more things. Because, you know, academics, we rely on funds, right, to carry out projects like this. Yeah. So, yeah, if there are funds, and Chaolong, this is what gives him the buzz, right? So, and I'm also feeling the buzz. So I'm very happy to work more on this if the funding is there to do it.
Yeah.
I think two things. One is the funding, the other is the community support. Like when we go to a broader region in Southeast Asia, we need to have people like you who support our research.
I'm sure after today, many will be stepping up. In fact, you have been invited to Malacca to do the research as well now. I've got people inviting both of you to go up to Malacca to get it done. So it's that dynamic now. A lot of people are pretty interested in this, and they are volunteering themselves for further research as well on that. Yeah.
That's
so
good to know.
And
if there are no questions, I have two questions to ask too, you know.
Okay, I think this is your avenue, Colin. Would it also be based on lifestyles or customs, as some Peranakans adopted children into their families, like Malays, when there's a child abandoned and orphaned, they just take them in and assimilate into our culture? So
yeah, I mean, I think our view is basically that while we all recognize that ethnicity has a role to play in our identity, I think we tend to lean more towards the cultural aspect of it, that, you know, what makes a Peranakan is not so much their ethnicity. Because when you talk about your own race, I mean, ethnicity has now been used almost symbiotically with race, right? When you ask any Peranakan Chinese what their race is or ethnicity is, they'll tell you. You know, for the Peranakan Chinese, they'll generally say, I'm Chinese. You know, for the Peranakan Indian, he's going to, or she going to say, I'm Indian. You know, so, but what makes a person a Peranakan, I think, has to do more with the culture that they invite and live. And while it's good to say that I've got mixed blood because my DNA profile tells me that, it doesn't necessarily mean that I'm Peranakan, because, you know, there are many people of mixed ancestry who do not declare themselves as Peranakan. They see themselves as Malay, or they see themselves as pure Chinese. You know, and we need to recognize that. Then how do you explain the situation where the self-declared 100% Chinese comes up and say, I am a Chinese Peranakan? You know, who are we to say that he is or he is not? You know. And so the genetic thing is very useful in just saying, okay, you're 100% Chinese, you're mixed Chinese Malay, you're even Indian in there, but that doesn't mean that you are not or you are Peranakan. I think that's very important to realize. So at the end of the day, it's whether you invite and leave the culture. You know, that's easier to defend. And having said that, I guess what's important for us to know is that we have to be very open about this whole thing. We don't want to be, we should not be attaching some exclusivity to whatever we have. We have to be open, and you are what you say you are, essentially that, you know. You can't say that, no, there cannot be any Peranakan Dutch, for instance. There cannot be any Peranakan Indian. There's only Peranakan Chinese. I mean, you can't even say that. And I don't think the association should be an arbiter for that. What the association should do is just try to bring some clarity to all these issues. But more importantly, not just to preserve and promote our culture, which we know very well, but to make sure that at least it goes on to the next generation, you know. You know, you have a whole new generation who come from Peranakan families, maybe on the maternal side or on the paternal side, who don't even know they are Peranakan, you know, and that's in all the letters, some of the letters that you read, even in the newspapers. The recent one being this young Chinese girl who has been raised as a Teochew, and she discovers that her grandmother or one of her grandparents is actually Peranakan, and she wants to find out more about her own lineage. you know, and it's also written about in Josephine Chia's book, exactly that, you know. So where do we go from here? And that's the whole purpose of having the convention, to go beyond what we already are. And for those of you who have yet to buy tickets, I'm going to put a marketing pitch here. Please go and buy, because you're going to find it fascinating. And so maybe I'll bring it back to a question that I have, you know. Why, in my conversations with both Roger and Chaolong, you say, hey, you know, that the female DNA seems to be very enduring. With the female genome, we can trace further back than we can with the male genome. Why is that so? Can one of you answer why is that so? You know what I mean? Because you're saying that when we trace generations back, a particular family's DNA profile, it is actually through the female genome that tells us that story that goes further back, rather than a male genome. Did I hear you all right saying that? Or am I wrong? Maybe I misunderstood you.
So let me try. The female evidence of female traces will appear in the mitochondrial genome, and the X chromosome is mainly female, but it also is found in male. So it's not so ideal to use that if you want to tell the sex difference. And then for the male traces, it will be in the Y chromosome, because females don't have Y chromosome. So when Chaolong looked at the Y chromosome traces and the mitochondrial genome traces, the best evidence of Malay ancestry was in the mitochondria, which is why he concluded that it was a female Malay ancestor. Whereas the Y chromosome, the Y chromosome looked quite purely Chinese and less Malay.
So when it goes to the information, I think both male genomes and female genomes, they provide us quite similar information. Females contribute slightly more because the X chromosome, female has 2 copies, male only has 1 copy. But the difference is very small, because majority of our genome is the autosomes. So I guess, yeah,
that's good. That's good. Gwen, any more questions relating?
We will just round out with one last question about percentage. What percentage of the samples have little Malay DNA? Does it show that there was a possibility of Peranakan based on self-declaration to be similar to pure Chinese, that is no intermarriage? Is there any way to determine that a person is pure, true blood Peranakan?
Yeah, well, that's quite a complex question. Let me try first, and maybe Chaolong go after. So, you know, when Colin asked the question, which some of you also had, as to whether our 177 was enough as a sample size, then it really is enough in so much as you want to see, and it really showed us the Malay admixture that we saw. But with 177, it really depends on whether you have the self-declared people have come up in larger numbers of those people who are really purely Chinese. So, you know, if you take another 177 and make it up to 300, then you might find that the pure Chinese are more than 10% of that group. You know, or you go to another 500 or 1,000 Peranakans, then you will find that there are more and more pure Chinese rather than Malay admixture. I mean, this is all hard to say. And I would say that, you know, for that reason, maybe a really large population study on Peranakans would be needed. But again, you know, it's all down to self-declaration, right?
Yeah.
Chaolong,
do you think that's a fair comment?
Not fully agree. So, because what you are saying actually have some indication that our sampling might be biased, right? What you are saying is that our current sample might have more participants who are
have the Malay admixture.
Who have Malay admixture, and those who don't have Malay admixture, they don't show up. And is there a reason that we have to believe this? I think when we broadcast, we broadcast to the community.
Yeah.
And if
okay, so this is where I don't have that confidence, Chaolong. So for example, right, we put the flyer out, and the people who come forward in response to the flyer might well be people who are much more possessing of the Malay ancestry, and they are people who like to volunteer for such things.
Yeah.
Whereas the people who are the Peranakans who are more pure Chinese are more withdrawn, right? So when they see a flyer, they ignore it.
Or the other way is that Colin, is there any patterns in the membership of the Peranakan Association? Because we send out the advertisement through the Peranakan Association. If your members, they are more like have more Malay ancestry, then that could affect. If not, then I think we are safe.
We don't really know because of the association. Our membership is about 2,150 today.
Yeah,
around there. And since 1964, we've always said the association is not going to be exclusive. It's going to be inclusive. We allow anyone to join us.
Yeah.
So it's very hard to say whether or not. But I think those who volunteered, when we went out to ask them, those who volunteered would generally be Peranakans. And whether they have mixed ancestry, nobody knows until now. Probably quite a large number are in that 177, and therefore they have all contributed to the aggregate percentage of 90% Chinese and 10% Malay, right? So I think most of them are probably mixed.
Yes.
But there are some I suspect who are pure Chinese, and we should not rule them out as not, as being Peranakan Chinese.
So by Chaolong's assessment or calculation, it was about 10% of the group that we recruited.
Okay.
Right.
That's quite a significant number, right? 10%.
Is it 10% that didn't have any Malay, large Malay ancestry?
Okay. Yeah. The aggregate number. That's interesting. Anyway, I think the time is almost up. I must really thank my technical team and Gwen for putting this together. Most of you, to the two of you and your team. I mean, and also our community for actually volunteering.
Exactly.
And I know of many of our friends and members who have said, I should have volunteered. I'm so sorry that y'all didn't, but I certainly would. I'm quite sure y'all would agree with me that this has been one very interesting session, and we should have more of this stuff. Yeah.
So Colin, before we hang up, can I put a plug to the research team, because
of course
we are very grateful for all the input, right? And then I think Peter is on the call here today, so thanks Peter for your help on this.
Yeah, we must. Hey, those of you here who are attending and who have volunteered, please get in touch with Peter Lee. Don't call Peter Lee as in L-E-E, okay? You come and complain to me and say, what do you do? L-I, Peter Lee.
Right. So, gentlemen, thank you so much. And we will be compiling the questions and the comments. And I think it was a very positive afternoon, a lot of wonderful comments. A lot of them are now volunteering themselves. Take me, Chaolong, take me, take my blood. I'm here for you, So they are volunteering and say, we are even willing to pay a fee for it. So many are stepping forward to wanting to be involved again if this research continues. But there
we'll
again, a shout out people from Myanmar, from Burma, from Philippines as well are interested to find out about their Peranakan heritage as well. So we hope there will be future sessions like this. And come back safe, Chaolong, to Singapore when this crazy COVID thing settles down.
Singapore.
Yes, I'm sure you miss the food here, Peranakan food.
Yes.
So
and the food there.
When Peter and I converse, we always start with food. So food is in our DNA.
Peter Lee was saying, so I said, no worries. I'm quite sure my wife Linda will cook for you all when you are here, Chaolong. You just come along.
Okay.
And provided we can we can go beyond 2, to bring them in.
So with that, I close this afternoon. Thank you, Roger. Thank you, Chaolong and Peter.
Thanks,
And,
guys.
To all our friends out there, don't forget the convention is on the 20th of November. Tickets are still available through SysTech. And last but not least, we have a feedback, QR feedback, which my colleague will be flashing quite soon, or is also on the chat. So kindly complete the feedback, and we're happy to hear from you. Yeah. Keep the emails coming, because I think Chaolong and Roger is fine with answering further questions. Yeah.
Yeah, okay.
On that. Yeah. So yeah, here you go. Your feedback matters. So please scan the QR code and let us hear from you. With that, stay safe, everyone, and take care.
Take care.
Good afternoon.
Bye-bye.
Bye.
Bye-bye.
Bye-bye.
Thanks, guys.
Bye. Bye.
Bye.